Hello, my name is Isabella Leonhard and my research focuses on conodonts, the earliest vertebrates bearing a mineralized skeleton, which makes them a great subject of the ongoing debate about their affinity, function and morphology. I'm going to talk about how to explore their feeding behavior, which is directly linked to the origin of predation in the earliest vertebrates of the fossil record. First, I want to talk about what conodonts actually are. They are one of the most controversial fossil groups, even though they are highly abundant microfossils from the Cambrian to the late Jurassic. By recent studies, they got placed somewhere in the vertebrate phylogeny between gnathostomes and cyclostomes, but their affinity is still an ongoing debate. They are regarded to be marine, eel-like, jawless vertebrates, possessing a feeding apparatus composed of phosphatic dental tools, which we call elements or just conodonts. Those elements are the commonly preserved part of the animal found mostly as discrete elements scattered in sediment. Soft body remains are rarely preserved, but the few body fossils found confirm their vertebrate affinity. Here we see the most famous body fossil ever found, the Granton conodont from the Granton shrimp beds in Scotland. They seem to have been predatory or scavenger feeding demersal animals with myomeric musculature, a notochord and big eyes. But still there's no verifiable homology between the elements themselves and vertebrate skeletal tissues. During the evolution, elements evolved from simple cone-shaped coniform forms, which we know very little about, to more complex and derived elements, which are at the same time the most studied and widely understood ones. In my research, I am focusing on coniform conodonts, which were highly diverse from the Cambrian throughout the Devonian, and encompass the order Paraconodontida, the Proconodontida, the Protopandodontida, and the Pandorodontida. I am focusing on Pandorodus from the order Pandorodontida from the Silurian, and on the even older Proconodontus from the order Proconodontida from the Cambrian. I hypothesize that these early coniform conodonts were actually active predators. Even though all conodont elements are highly diverse in their morphology, they all have some unique features in common. They are all divided by histological studies into paraconodonts and euconodonts. Euconodonts are also called real conodonts or just conodonts, while paraconodonts seem to be the ancestral species. Even though euconodonts are highly diverse in their forms, they all consist of two different types of tissue: the dentine-like basal tissue and the overgrowing enamel-like crown tissue. The basal tissue is completely homologous to the paraconodont element, which is the reason why euconodonts are very likely derived from paraconodonts. Euconodont elements grew through accretion of new lamellar tissue in an appositional way, which is actually different from other vertebrate teeth. This mode of growth gives direct insight into the animal's ecology by preserving tissue damage and wear fractures, as well as chemical changes between the different ontogenetic stages. Here we see a schematic drawing of tissue damage and subsequent repair by overgrowing crown tissue. Phil Donahue proposed in 2001 that the crown tissue seems to be an adaptation which evolved under evolutionary pressure to stiffen the whole element and to protect the basal body. A tooth-like function is therefore strongly suggested for all conodont elements. But their diversity in element form and shape indicates an enormous range in their feeding ecology. A food processing function, especially for the most studied complex conodonts, is therefore very likely. Conodont elements are commonly arranged in a bilateral, symmetrical, multi-element feeding apparatus. To explore their feeding behavior, it is essential to know how these elements were arranged. For complex conodonts, multiple apparatus reconstructions and functional model exist, while there's less evidence for coniform conodonts. Why? Because their rarity and their simple morphology leaves less space for interpretations. The reconstructed feeding apparatus for complex conodonts can be broadly broken into two parts. The S-array, which consists of ramiform elements located towards the front with a grasping function, and the platform elements, which are located in the throat with a slicing and grinding function. Coniform conodonts did not bear platform elements, which makes the reconstruction even harder. But thanks to Jig, Dragon and Sansum, a complete feeding apparatus reconstruction of the species Pandorodus onychostatus has been around since the 80s, which based on probably the most famous fused cluster ever found for coniform conodonts, the Podolina cluster from the Ukraine, which firstly got described in 1986. Discrete elements of conodonts scattered in sediment do not tell us much about how they were arranged in an apparatus, but fused clusters and bedding plane assemblages do. Sadly, there are fewer clusters found for coniform conodonts than for complex ones. But nevertheless, the apparatus reconstruction from Sansum can be applied to all other coniform conodonts. In total, the Pandorodus apparatus consists of 17 laterally furrowed elements, divided into 6 different morphotypes. A functional interpretation was added by Duncan Murdock in 2013, who divided the elements into those with a grasping and those with a cutting function by using inferred mechanics. In addition to other sources, I am using this apparatus reconstruction of Pandorodus to test the hypothesis that the early coniform conodonts, Proconodontus and Pandorodus, were active predators. My results may indicate evidence that predation already existed among the very early conodonts. By BSE imaging, I want to test if there are any veer fractures or tissue damages present, and if so, does their location fit within the functional model reconstructed for Pandorodus? Secondly, I will do EDX measurements to test if the strontium content changes during the different ontogenetic stages, and if so, do these changes indicate higher trophic level in adult age? Finally, I want to compare tissue damage and changes in strontium content between Proconodontus and the more derived Pandorodus to reconstruct what functional adaptation was driving differences between these both genera. The question that needs to be answered here is, do we see any increased specialization within the apparatus or the elements within the very early conodont evolution? Here I should point out that because of the current situation of the coronavirus, I am not able to give you any lab results yet. Besides BSE imaging and EDX measurements, I am using synchrotron scans of Proconodontus, which got imaged at the Tomcat beamline in Switzerland. This 3D data gives us a lot of information about the internal structure of specimens without actually destroying them. With help of 3D modeling, it is possible to identify different morphotypes and to generate as many cross-sections as you like. My project is designed to answer important questions in paleontology. It will not only provide evidence that predation already existed among the very early conodonts, but more broadly will also help to understand the origin of predation in the whole vertebrate lineage.